When we think of regenerative medicine, we think of a complex process. Removing cells from the body, re-engineering genetic structures, returning the modified genes. At Frequency, we are creating a simpler way to unlock the potential of regenerative medicine. Within our bodies lies the innate biology necessary to generate new healthy cells, like those we were born with. Some of these cells are active and continue to repair and regenerate tissue over time, but others are dormant, sitting idle. Consider hearing loss. Throughout evolution, nature assumed that the 15,000 auditory hair cells we're born with are all we'll ever need to hear. But evolution didn't anticipate airplanes, subways, sirens, and headphones. As the sensory hair cells of the inner ear die away from noise or disease, we lose our ability to hear. But the dormant progenitor cells that generated those hair cells are still there in the cochlea. At Frequency, we believe we've discovered the pathways where small molecules can activate the progenitor cells in the cochlea to regenerate sensory hair cells. Using this approach, we have observed the first ever statistically significant hearing improvements in clinical trials, as we seek to deliver the first regenerative therapy to reverse hearing loss for millions of patients. We believe the same regenerative approach can reverse damage caused by multiple sclerosis. In multiple sclerosis, the myelin-insulating nerve cells break down, destroying connections, leading to lost motor skills and disability. The precursor cells that originally formed these myelin sheaths are still present in the brain, and like in hearing, we believe they can also be activated. Following specific pathways, we have uncovered a novel target, and our aim is to develop a therapeutic that activates these precursor cells to regenerate healthy myelin and restore function. Our approach is straightforward. We're using small molecule therapeutics to safely activate a person's innate regenerative potential within the body to restore function and treat the underlying cause of disease. Frequency Therapeutics, leading a new evolution in regenerative medicine.
Good morning. My name is David Lucchino. I'm the CEO of Frequency Therapeutics, and I want to thank you all for joining Frequency's first R&D event. Today, we'll be making forward-looking statements. I would refer you to our recent SEC filings for additional information. Since our company started, we have been working to develop a new category of regenerative medicine, using small molecules to activate a person's innate regenerative potential within the body. Unlike more complex cell and gene therapies, this is a simple approach with huge benefits and potential. Today, our team is going to show you significant progress in our lead clinical program for hearing restoration. We will unveil 2 new, very exciting programs as we grow our pipeline of regenerative therapies. For those who have followed our story, Frequency is leading the field of hearing restoration with the first potential therapeutic for acquired sensorineural hearing loss. But saying we are leaders and showing we are leaders are 2 different things. Today, you will see all the data from each of our FX-322 studies. These data, when pooled together, make a clear and compelling case that FX-322 has a clinically meaningful impact on improving speech perception, the key unmet need for people with hearing loss. We will then outline how these data have informed a new, recently initiated Phase 2b study designed to focus on where we have seen the greatest response from subjects. We believe that by focusing on patients that have the best response, we'll be able to show the greatest separation from placebo and therefore advance a hearing therapeutic towards commercialization. But FX-322 is just the beginning. Over the past few years, we have consistently been asked, what if we were able to get drug deeper into the cochlea? Today, you will see preclinical data from a new hearing program, FX-345, that does just that. This potentially gives us a greater ability to treat more and different kinds of individuals with sensorineural hearing loss, and we are excited to move 345 into clinical studies. We've also been asked, what if we extended our approach to other degenerative diseases. Today, you will also see exciting data from our preclinical program for remyelination and multiple sclerosis, where we have discovered a novel target as we work to advance a new restorative treatment for this debilitating disease. These are the first data for what we believe may become a highly differentiated offering for patients with MS. To take you through our progress, we have an impressive group of speakers. First, Dr. Robert Langer, a co-founder of Frequency and an MIT Institute professor, will discuss the complexity of pioneering a new category in regenerative medicine. Following Dr. Langer, leading members of our clinical team will walk you through how the clinical data from our combined FX-322 studies today show a clear pattern of improvement in speech perception, how our new 208 study is enrolling a population where we believe we are most likely to see a clinically and statistically meaningful benefit, and how we now have aligned with the FDA on speech perception as the primary efficacy endpoint for our studies moving forward. These factors support our belief that we have a very promising candidate to be the first approved therapeutic for hearing restoration. We're also thrilled to be joined by two distinguished key opinion leaders, Dr. Sumit Dar of Northwestern University, a leading auditory scientist, and Dr. Steve Targum, a world leader in clinical trial design. They will provide context for how our clinical data aligns with the pathology of the inner ear and the strength of our new FX-322 study design. Next, our research team will walk you through two significant areas of pipeline expansion: a new hearing restoration program and our new program for remyelination and MS. This is a key inflection point for our company as we work to build a robust pipeline based on our core science. It is a time where I believe we are creating important new medicines for patients and tremendous value for our shareholders. Following our presentation, we'll be hosting a Q&A session with all presenters, and we look forward to hearing from you. This has been a remarkably productive period for Frequency, one that's been all about execution and pipeline expansion. We have a very aggressive program, though I believe we have the team and resources to advance these programs to millions of individuals in need of new treatment options. Thank you very much for your time today. I will now hand it to Dr. Langer for his remarks.
I've had the pleasure of working with a number of biotech companies, in fact, starting some and being on the boards, either scientific advisory board or board of directors of others. Some of those include Genentech, Alkermes, Momenta, Moderna, Alnylam, and all those companies have had real breakthroughs. But part of what's enabled them to be successful has been the ability to target drugs to the exact location of the body for optimal performance. And if you look at the companies I mentioned and many, many others, getting through this challenge of getting to the target, that has allowed them to demonstrate the potential of new modalities and breakthrough approaches to treating all kinds of diseases. And almost out of the gate, Frequency was able to translate research actually that was developed in our lab along with Jeff Karp, who's now a Harvard professor and a former postdoc of mine, where we discovered small molecules, and Frequency was able to develop a novel gel that they put these small molecules in, and they were able to use that system to enable the drug to enter the cochlea, the delivery system. Now, as I'm sure everyone knows, the cochlea is very challenging to penetrate, and I've been really excited to see this work validated by the hearing signal that's been observed across multiple different studies. Also, I'm sure everyone knows that drug discovery is never a straight path. In fact, every company I've been involved with has had ups and downs. That's the nature of biotech. Things are unpredictable. And Frequency had confounding data in its phase 2A study of FX-322. But while disappointing, I believe these results have really provided the Frequency team with new essential insights at a really critical time in their development. I believe the company has responded exceptionally well. And today you'll hear compelling new data and insights, including important new advances in cochlear drug delivery that I believe will further Frequency's leadership in hearing restoration research and development. I'd now like to hand this over to Carla Bell, who's Frequency's Chief Development Officer, and he's going to discuss the company's ongoing work to develop the first therapeutic to potentially restore hearing. Thank you very much.
Thank you, David and Bob, and welcome everyone to the first R&D event. I'm Carl LaBelle, the Chief Development Officer, and I want to start by saying how grateful I am to be part of the Frequency team that's leading the hearing restoration field. Today I'll summarize key translational elements of the FX-322 development program, and in particular our experience in the three largest etiologies of acquired sensorineural hearing loss, such as noise-induced hearing loss, sudden sensorineural hearing loss, and age-related hearing loss. We were the first to demonstrate a pharmacokinetic-pharmacodynamic relationship for FX-322, where we showed that therapeutic concentrations of FX-322 in the human cochlea were associated with statistically significant improvements in hearing function. This historical milestone was achieved in parallel with us identifying a positive signal in hearing function that was manifest as improved speech perception in patients with permanent acquired sensorineural hearing loss. These improvements in speech perception were sustained in some subjects for almost 2 years, suggesting a potential disease-modifying benefit. Furthermore, we have uncovered that some patients may respond at different times, important learnings to the overall program. Our regenerative approach is unlike any other being taken in the hearing loss space. Our focus is on hair cell development, where we decode the signals that drive these cells, and we modulate those signals with combinations of small molecules. We demonstrated this work in vitro in cochlear progenitor cell cultures, showing that we could generate all the cellular components of a functional sensory hair cell, such as formation of hair bundles, functioning ion channels, and synaptic signaling with neuronal connections. It is the loss of these sensory hair cells that is thought to be the primary consequence of acquired sensorineural hearing loss. As our hearing biology team continued the translational work, we were able to show in a murine noise-induced hearing loss model that FX-322 could regenerate inner ear sensory hair cells, as well as restore hearing function. Here on the left is the cochlea from a vehicle-treated mouse with a noise-damaged ear that is missing sensory hair cells, evidenced by the loss of the pink-colored sensory hair cells. On the right, you can now see a cochlea from a noise-damaged ear that was treated with FX-322, showing increased numbers of sensory hair cells. This in vivo proof of concept work is in press at the Journal of the American Academy of Audiology. Key to drug development is the ability to show that enough drug gets to the tissue and targeted site of action. We published work earlier this year in one of the premier journals in the field, Otology and Neurotology, where we demonstrated that sufficient concentrations of FX-322 get to the human cochlea. Here you can see an illustration that FX-322, shaded in orange, concentrates mostly in the base of the cochlea after intratympanic injection. In the base of the cochlea is where the high-frequency signals are processed, and this is important because humans typically will start losing hearing function first in this range. Later in the program, Chris Luce will speak in greater detail on the cochlear pharmacokinetic profile of FX-322. Reflecting on the clinical program that we've been conducting since 2018, we have amassed a considerable database on how FX-322 performs in patients with acquired sensorineural hearing loss. In this program, we have completed 4 studies that involve nearly 200 patients. Importantly, FX-322 has continued to exhibit a very favorable safety profile. Now let me walk you through each of these studies separately and share with you the learnings. The 201 trial was a single-dose, double-blind, placebo-controlled safety study in patients with either noise-induced hearing loss or sudden sensorineural hearing loss in the mild to moderately severe range. Patients were dosed with FX-322. They returned to have their hearing tested several times over a 3-month period, and it was in these follow-up visits where we first observed FX-322 was associated with statistically significant improvements in speech perception. What was also notable is that when we invited some of the patients that had shown speech perception improvements to get retested 1 to 2 years later, several of them had maintained their improvements, which is a remarkable finding for a sustained effect following a single injection. Given that FX-322 is maximally formulated, we decided to test for different FX-322 exposures by giving multiple injections. Here you can see the 202 study, which tested the same patient population where we observed the speech perception signal in the 201 study. I will speak to the results of this study a bit later in this section. At this stage of the program, we were interested in understanding what were the boundaries of where FX-322 might provide benefit. And here we show a grid that is organized by severity along the top and etiology along the left side. In our FDA interactions, it was understood that to seek a broad indication, such as for the treatment of acquired sensorineural hearing loss, we were going to have to study all etiologies and all severities. Therefore, the next two studies were designed to enable us to work around this grid and to explore safety and look for a signal in various patient populations. The 111 study was a single-dose open-label safety trial in patients with any etiology in the mild to severe range, and they were dosed with FX-322 under two different administration conditions in one ear, and the untreated ear was used as a control. This was then the second trial where we observed that FX-322 was associated with statistically significant improvements in speech perception. Interestingly, we invited all patients to get retested 8 to 12 months later. Several additional patients showed improvements, which provided us the insight that some patients may respond at different times. The next trial was the 112 study, single-dose, double-blind, placebo-controlled safety trial in age-related hearing loss patients that were 66 to 85 years old. It is important to note that this older population study did not have any evidence of noise-induced hearing loss or sudden sensorineural hearing loss. We did not observe a robust speech perception signal in this older patient population, but did note that one patient had statistically significant speech perception improvement. The scale of the FX-322 clinical program has enabled us to pool the 3 single-dose trials to look for patterns in the data and give us important insight into the characteristics of responders. I'm now going to show you a couple of ways that we've been evaluating pooled data. The first pooled data analysis we performed looked for how many patients show at least a 10% absolute change from baseline in speech perception, which is one of the responder definitions. In this graph, the y-axis is the change in the number of words correct from a 50-word test. Here you can see highlighted the plus minus 10% absolute percent change from baseline, or plus minus 5 words, which is the cutoff for clinically meaningful speech perception improvement. And Kevin Frank will speak more on this topic. Now, when we pool the data from the 3 single-dose trials, you can see changes in FX-322 treated patients that clearly exceed the threshold for meaningful improvements, while a small fraction of placebo-treated patients or untreated ears land above the threshold. In fact, several FX-322 responders showed at least 10-word improvements or a 20% absolute word increase, while some had a 40% absolute word increase. Another way that we look at the pooled data is by using a responder definition of the number of subjects that exceed the 95% confidence intervals from well-established historical standards. Looking at the data in this way, you can see that 14% of all FX-322 treated patients exceed the threshold for improved speech perception, and how the placebo-treated patients and untreated ears are consistent with the historical literature standards. Now, using this same responder definition, let me bring you back to the 202 study and discuss how this multi-injection study compared to the single-dose trials. When we compare the proportion of placebo-treated patients and untreated ears, we see that 15% of placebos and untreated ears exceeded the 95% confidence intervals, and the 202 study is a clear outlier. The biggest difference between the 202 study and most of our single-dose studies is that we only collected a single baseline measurement. As we have shared, there is evidence that baseline speech perception scores were not consistent with historical scores for many of the subjects on this trial. Therefore, if the baseline value is not accurate, you're always comparing against baseline, the study is likely doomed for failure. Steve Targum will speak specifically to this element of patient bias later in the program. The hearing signal that we've observed is manifest as improvements in speech perception, meaning study subjects can understand more words and experience better communication. It has also been well established, as we described in Otology and Neurotology paper, that providing better function, perhaps by restoring sensory hair cells in the highest frequencies where FX-322 is localized, may enable better function in the mid-frequency speech range. This is possibly related to restoring some tuning and filtering function in the base of the cochlea. These are concepts that will be discussed in more detail by Sumit Dar. Further support for the relevance of speech perception as the most impactful component of acquired sensorineural hearing loss was broadly discussed by patients at an FDA patient-focused drug development program that was led by the Hearing Loss Association of America. When asked about the two top needs for a drug or a device, patients were most hopeful for improved hearing and hearing restoration, and their chief complaints were trouble hearing in background noise and sounds being muffled. It is clear from this event that patients want to hear more clearly, and speech perception is the primary unmet medical need. Shortly after the PFDD meeting was held, we reviewed the results of the 201 and 111 studies with FDA at a Type C meeting. It's fair to say that early assumptions of the hearing loss field were to expect audibility improvements with FX-322. We are pleased to say that we are fully aligned with FDA that the primary registrational endpoint for our trials will be a speech perception measure, and that the next study that I'll share with you has been vetted with FDA. We believe that the alignment on endpoints during phase 2 of development reduces the risk to the program and increases the probability of technical and regulatory success. Lastly, we've been developing a proprietary patient-reported outcome measure that we call Radial, in collaboration with PRO experts as part of our clinical development program to collect data on the impact of FX-322 on patient daily activities. Most importantly, Radial is being designed by patients with acquired sensorineural hearing loss. I hope that this gives you helpful perspective on how we view the FX-322 program. We've developed a novel drug-drug combination that regenerates sensory hair cells and cochlear function preclinically, that we've been successful in demonstrating first-of-its-kind translational data from bench to clinic, showing hearing improvements in 2 independent trials, a favorable safety profile from 4 trials in nearly 170 treated patients, and that we have alignment with FDA on endpoints that mean the most to patients. All of the learnings from the single-dose trials have informed us on the 208 study design that I'll discuss later in the program. With that, I would like to introduce Kevin Frank, who will summarize for you all of the single-dose data.
Thank you, Carl. Before I took my role at Frequency Therapeutics just less than a year ago, I ran one of the largest audiology clinics in the world at Mass Eye and Ear, Harvard Medical School, where I provided direct care to patients with hearing loss. I worked with patients as they lost their hearing, and I measured changes when we applied various device interventions and surgeons performed their procedures. Clinicians like me widely agree that an absolute change of 10% in a speech perception test is meaningful and has a noticeable impact on patients' lives. On a clinically standard 50-word list, that means 5 words. If you start at 12 words, getting to 17 is clinically meaningful. Today, I'd like to share the impact 10% improvement in speech perception can have on patients' lives and how we apply additional statistical rigor for the purposes of drug registration clinical trials. Early in the presentation, Carl showed you individual data points from all subjects who received a single dose of FX-322 and completed the 201, 111, and 112 studies. Here, I'll provide more detail and show you even more. First, a note about the scale. When we talk about 10% or 5-word improvement, we always talk about an absolute change. Why do clinicians agree that 5 words is meaningful? Because it can lead to changes in their treatment recommendations. At 35 words out of 50 in quiet, this person has moved past the phase of wondering if she has hearing loss. She's waited years to get her hearing tested and is faced with the decision of what to do about it. In quiet places, she can get by with context, but in noise, she's lost. A 5-word improvement to 40 out of 50 words delays the need for professional intervention, as she may get by with deliberate work and home accommodations. A 5-word decrease in speech perception to 30 out of 50 necessitates a professionally fit hearing aid, forcing her to confront a variety of powerful barriers and stigma. At 20 words out of 50 in quiet, even with well-fit hearing aids, this person struggles. Home and work communication are both difficult, and he is at risk for depression due to the impact this has on self-confidence and independence. He's on the border of cochlear implant candidacy. A 5-word improvement to 25 words out of 50 brings back some of the conversation, phone use, and delays surgery. A 5-word decrease to 15 words out of 50 makes him a clear candidate for a cochlear implant. On the x-axis here, we have a baseline score, and on the y-axis, we have the post-dose score. The diagonal would be equivalent performance. 5 words, or 10%, in this direction is a clinically relevant improvement. It'd be unexpected for a score to go up because sensory hearing doesn't improve. It'd be less unexpected for a score to go down, as unfortunately, we all head that direction eventually. There's an additional tool relevant to measuring differences in speech perception that adds statistical rigor when describing changes, which is particularly helpful in clinical trials. The gold standard Thornton-Raffin statistical test defines this curved boundary of 95% statistical significance. Note how it's thicker in the middle and narrower in the ends. Outside this boundary, we have confidence, and for most of the curve, this boundary is further beyond the 10% clinically meaningful border. As I said, with sensory hearing loss, we all head down on this graph, as our cochlea takes on a lifetime of damage. What's remarkable about our results so far is that we see clinically and statistically meaningful shifts up. You've also heard about the other way data go up, through bias expressed through reduced baseline scores and inconsistent effort. With this view, we can observe if bias confounds our data. We've shown these data in the Otology and Neurotology publication, but I've added hearing audibility measured by pure tone average indicated by color. For illustration, the arrows show that one subject with moderately severe hearing loss started at a baseline of 20 words and improved to 39. The 4 subjects above the line have demonstrated both clinically and statistically meaningful improvements in speech perception. 3 of them double their score, with improvements around a 20-word change. That's 40% on an absolute scale, which would make a remarkable impact on the subjects' lives. Note the 4 subjects with moderate hearing loss who received FX-322, the gray dots. Turning the graph to level baseline performance, let's focus on how the subjects with moderate hearing loss line up. This first one had a 4-word increase that was not clinically meaningful and was never counted as a responder. Another had a 9-word or 18% upward response, almost double what's clinically meaningful. But now, with this view, you can see not over the 95% confidence limit on the line. We've decided to be stricter and not count subjects like this who are above the 10% 5-word clinically meaningful improvement, but not also above the statistical mark, because we don't have confidence in the difference. The same 9-word improvement level in another subject to the left was both clinically and statistically better, and we mark the subject as a responder. Equivalent number of word improvement can have different statistical meaning depending on the baseline. Finally, look at this responder with more than a 20-word, 40% improvement. Confidently meaningful and quite exciting. We've never seen kinds of improvements before like this in pharmaceutical treatment of the cochlea. Most of the subjects who showed an improvement in words and quiet also did so for words and noise. To reiterate, the purpose of looking at the data compiled this way is to further refine the definition of a responder for future study powering and to provide you with transparency showing every subject's data point. We apply a higher standard of statistical difference on top of what's meaningful in a way that's consistent with best clinical practice. Looking only at the 201 data now, with untreated and placebo ears, this view doesn't add much, as most of the subjects who are clinically meaningful are also statistically significant. 201 was a small study, an encouraging one, but a small study nonetheless. Now, let's add the 2 other single-dose study numbers 111 and 112. Combined with 201, you can see 71 subjects treated with FX-322 who completed the studies. 13 with placebo, and 84 untreated ears. Now we see the benefits this view provides. The first benefit is to see the distribution of the data by group. The untreated ear group has the most data points. The shape is normal, wide in the middle and narrow in the ends. We focus on subjects going up on the graph because, as I said before, we don't expect subjects to improve without treatment. The 2 subjects out of 84 whose untreated ears exceed the threshold is 2.4%, what statistics would predict. The placebo group has none. the treated group looks markedly different than the other two. It has a long tail in that upward direction that can't be explained by chance. This gets us all very excited here at Frequency. We've studied details about every visit of each of these subjects and observed characteristics that influence our future trials and help further separate the behavior of the FX-322 group from the placebos. Contrast this with the 85 subjects who completed the 202 study. The 85 untreated and 18 placebo ears show a much higher percent, 11 and 37% respectively, or 15% combined, as Carl pointed out, than the 2.5 we'd predict statistically and what we observed in single-dose studies 2% of the time. We believe this is due to the inconsistent baseline bias before dosing, as Carl mentioned, and why we don't pool the 202 study with the others. This slide is showing you all of the words and quiet speech perception data we've measured so far from our 169 subjects who've completed our studies in both ears. When we examine our responder rate by etiology and severity, we can see where active subpopulations reside, up to 40%. The size of the bubble reflects the number of people tested in each segment, and the color reflects the percentage of these people who responded beyond the 95% confidence interval. To illustrate, compare the 27% responder rate in the moderate hearing loss group with sudden hearing loss with a 7% with presbycusis. Similar number of people tested, different responder rates. These subpopulations guide us in our 208 clinical trial strategy to separate FX-322 from placebo, as Carl will share with you later. We're not shutting the door on spaces without robust responses. To me, these early data from small exploratory trials are very encouraging. Each of our responders would have experienced a life-altering improvement, and we're beginning to recognize characteristics about them. We've pushed these subjects up on the graph, something no one's been able to do before. As a clinician, I wouldn't hesitate to recommend a drug like this. Now, I'd like to introduce Dr. Sumit Dar. I've known Dr. Dar through his leadership, his knowledge, and his collaboration for decades, and I'm thrilled he's able to address you today. Dr. Dar will discuss high-frequency hearing ranges, an area critical to speech perception, but not part of a typical audiology workup, because there's nothing to treat in that area right now, and other important points around cochlear heterogeneity. Thank you very much.
Hello, everyone. My name is Sumit Dar. I'm the Hugh Knowles Professor of Hearing Science at Northwestern University. I am happy and delighted to speak to you today about cochlear pathology and the impact of high frequencies on speech perception. Here's the disclosures. The only one that you need to pay attention to is the top line, where Frequency had me be part of a panel, and they shared all of their past data and plans for future clinical trials. And from that, we started a discussion about my work and their work, and three big questions emerged that I'm going to try to answer for you today. The first big question is, why treat the high frequency region of the cochlea? As you know, that's the region of the cochlea that Frequency's treatment targets first. Well, there are good reasons to do so, and I'm going to try to talk you through that from a data set of almost 1,000 individuals from my lab. We've been publishing these data over almost the last decade. What you're seeing here on the y-axis is how loud or how big a sound has to be before it's barely audible. Along the x-axis, the frequency of the signal. This vertical red line here marks where we stop testing clinically today. So anything beyond that is essentially experimental data that we are trying to make mainstream. The flat blue line up top is the reference point. This is the average hearing of 10 to 14-year-olds in this data set. And then each subsequent line after that marks how much hearing you lose for every 5 years of age. This dashed red line shows us the 20 dB mark, which is considered by many to be a significant amount of hearing loss. As you can see, it doesn't take long. By the time we get to 20 to 24-year-olds, we have 20 dB of hearing loss, clinically significant amount of hearing loss. Well, Why do we not worry about it? Because we don't test at the high frequencies, we test in these mid frequencies, and we carefully have chosen individuals who have clinically normal hearing to demonstrate there already is a lot going on in the base. The next interesting thing is that the biggest drop in hearing happens between there for 20 to 24-year-olds to here in 44 to 49-year-olds. Well, then things slow down. But when we are measuring age-related changes in hearing in these mid-frequencies, we essentially see the fumes of what has already happened in the base. So this is a data set that tries to identify the effects of just aging. If we had other toxins in place, such as noise or toxic drugs, middle ear infections, what we would see is that these mid-frequencies here would also drop, and this notch would get widened. So it is clear from these data that it is critical to treat the high-frequency regions of the cochlea, because that's where most of the common forms of hearing loss start and spread into other parts of the cochlea. The next big question I wanted to answer is, how does high-frequency cochlear function influence speech perception? Let me start with the answer that it does, and then share another data set of almost 1,000 individuals. Again, this we have published recently. What you're looking at here are hearing thresholds, as you've seen before, not referenced to any age group, but just a decline in hearing with every 10 years of life. Below here are otoacoustic emissions, distortion product of otoacoustic emissions. These are sounds that are generated within the inner ear as a part of the hearing process. We put small microphones down people's ears and record them. What you see that the cochlea, or the inner ear of the 10 to 21-year-olds, generates most of these sounds. And then as you get older, the amount of sound generated by the inner ear drops, marking a deterioration of function in the cochlea. You also notice that, again, the biggest changes or the most damage is seen in the high frequency regions of the cochlea beyond the red line where we stop testing. Again, emphasizing the first point, it is important to test these regions of the cochlea and treat these regions of the cochlea, but then ask the question, does damage in these parts of the cochlea affect speech perception? Yes, it does. So in this data set, when we did a principal component analysis, we found that the OAE levels between 12.5 and 16 kilohertz high frequency regions had a significant impact on speech perception scores. So this is not evaluated routinely because folks don't pay attention to the high frequency regions, but in our hands and hands of others, this is becoming more and more of an important part of the cochlea to evaluate and treat. And speech perception is the right measure to use because most people who walk into a clinic looking for treatment for hearing loss walk in because they can't understand speech. And also, speech perception has been a long-standing gold standard in all treatments of hearing loss, cochlear implants, hearing aids, etc. So this is the right gold standard to use. Then the final question to answer is, should we expect to see heterogeneity in treatment outcomes? Well, heterogeneity in treatment for hearing loss has been a reality for as long as we've tried to treat hearing loss. Decades of data from cochlear implants and hearing aids show this heterogeneity, and it's likely driven by varying amounts of damage, where that damage is across individuals, and varying rates of plasticity in the auditory system. These are things hard to measure in the human system because it would take invasive ways of doing so. So we assume a lot, but don't know precisely where the damage is or how much the damage is. This leads to heterogeneity. So what is the impact of this heterogeneity on clinical study planning? Well, you take a group of individuals in who on the surface have similar hearing losses, but underneath that, there could be wide variety of etiologies and duration of hearing loss. The Frequency's approach of targeting a broad audience, a broad population with various etiologies and pathologies in the early studies was really necessary to identify the target population. And then the steps they've taken to put further controls in place for the next steps are, again, the right next steps to pursue. So what are the takeaways from the three questions I've tried to answer? First, treating high frequency region of the cochlea is critical. Second, the high-frequency region of the cochlea does influence speech perception, and it's becoming more and more clear as we focus on that region of the cochlea, not only in my lab, but in various labs. And then heterogeneity of response is a reality of all treatments for hearing loss. I am very hopeful that the controls that Frequency has put in place for the next trials will help us learn a lot more and find the right target population. And this is something that needs to be done. There are a lot of people who need this help, and I am so happy that Frequency is following this. Thank you so much.
Thank you, Sumit, for providing your insight on the importance of high-frequency function as it relates to speech perception. I hope the presentations so far have reinforced for you that for future trials, we will be focusing on patient populations that show the greatest separation from placebo. Given the summary that Kevin showed you on all of our trials, we emphasize 3 key principles for the design of the FX-322-208 clinical trial. First, we focused on the patient populations where we've seen speech perception signal in prior trials. Next, our intent was to design the 208 trial at a scale that can confirm efficacy. And to achieve that goal, we used a pooled data strategy. Lastly, we incorporated design principles that have been tested in other disease areas that can mitigate bias that we observed in the multi-injection study that use only a single baseline measure. In Kevin's talk, he shared with you this bubble chart that provides a grid across sensorineural hearing loss severities and etiologies that have been studied with FX-322. You can see that there are certain populations of patients, like moderate to moderately severe noise-induced hearing loss and sudden sensorineural hearing loss, that shows around a 30 to 40% response rate to FX-322. The orange circle indicates the focus that we have for the 208 trial with respect to the patient populations. But you should note that the circle trends in both the high milds and low severes, and these groups are also included in the 208 trial. Therefore, our pooled data strategy has led us to focus on sudden sensorineural hearing loss and noise-induced hearing loss patients in the high mild to low severe range. This pooling strategy indicates that 25% of subjects treated with FX-322 show improvements in speech perception. As we discussed previously, 0% of placebo and untreated ears show improvements, but we have assumed a 5% of placebo subjects would show an improvement, and therefore our treatment effect is targeted to be 20%. Thusly, we need a sample size of 112 subjects at 80% power to detect this 20% difference at a significance level of 0.05. We will be recruiting 124 subjects into the study to account for potential patient attrition. We have employed several new design elements to mitigate bias, such as lead-in phase with multiple visits to assist with observing a greater separation between FX-322 and placebo. Here you can see that the 208 trial has 3 lead-in visits to establish a sound baseline measure based on consistent test results. Other design elements, such as masking of both patients and sites, further mitigates potential bias, and a level of audiology surveillance is built into the study such that all test sessions are recorded. Finally, the lead-in phase is used to disqualify patients with poor test consistency. Steve Targum, who will speak next, will dive deeply into these items as they relate to our program. Therefore, the 208 study will recruit 124 patients. It is equally balanced with 62 patients per cohort and adequately powered with a primary endpoint of speech perception. I am pleased to say that this study is underway and patients have been randomized to the trial. We will provide timeline guidance to you when we have a good understanding of the enrollment rate. We are confident that these design components bring in a level of clinical quality control that is unparalleled by any other trial in this hearing loss space. To summarize, the signal that we have observed in 2 trials aligns with the outcome measures that have been agreed upon with FDA, as well as what matters most to patients as described at the PFDD. Our large clinical database has enabled us to pool data and better understand responder profiles, and we've captured important learnings from all of our trials that have been incorporated into the 208 study. Taken together, we remain confident that the 208 Phase 2b trial is optimally designed to confirm FX-322 improvements in speech perception. With that, I would like to introduce Steve Targum, who will discuss his experience with placebo and bias issues in clinical trials and how we have addressed them in our 208 study design.
I'm Dr. Steve Targum, a clinical consultant at Frequency Therapeutics, and I'll be addressing placebo response in clinical trials and explore some of the best practices that Frequency will employ in their 208 study. This slide reveals my disclosures, and as I noted, I'm a consultant to Frequency Therapeutics specifically for the purpose of their design of this next trial. What I'd like to examine is the challenges that exist in all clinical trials, and a key issue, of course, is reliability. And some of the challenges that are inherent to any clinical trial, particularly in CNS, are misplaced enrollment incentives, either by the staff, who are only paid to include patients, not reject patients, and by subjects who are obviously motivated to get into the trial. Some subjects will exaggerate their symptoms so that there's a score inflation that makes it seem as if their deficit is worse than it really is. A second factor is expectation bias. People enter a trial because they want to get better. They consent because they believe that they may benefit from their participation in the trial. This type of expectation may also stimulate a response. We see it even in studies where we presume the measures are objective, such as rigidity in Parkinson's studies, which may improve simply because the patient believes they will get an effective treatment. The third factor is pre-randomization symptom fluctuation, and that's simply because symptoms don't always stay the same. And yet it's important that we have a relatively stable baseline measure because that is, as I mentioned before, the contingency variable upon which the outcome relies. And finally, another factor, but certainly not the only factor, is ratings precision and consistency. Raters are human beings, as are patients, and they may be consistent or inconsistent between visits. Subjects may be cooperative or uncooperative between visits. All of these factors may affect the outcome. And these same principles that affect CNS trials apply across all clinical trials that use placebo controls, including hearing loss. The failure to manage these factors obviously can impede signal detection and obscure the results and make it difficult to determine if the drug is really effective. So how does this information apply to Frequency? Well, Frequency took a careful look at their 202 study and learned that some of the problems that we see in CNS studies existed in their study as well. First, an inflation of hearing deficits at the screen. Some patients exaggerated or misrepresented their symptoms in order to gain entry. They underreported the number of words they could understand from the word recognition test in order to achieve study eligibility. Another issue was open communication about eligibility criteria. Some clinical trial staff actually shared with subjects the reasons that they were excluded from the trial. And Frequency themselves vocally communicated the requisite hearing deficits necessary to enter the trial and the intended primary endpoint. Some of these subjects who knew the reasons that they were excluded and knew what the required entry criteria were, shared this information on social media platforms that are routinely used by subjects with hearing loss. Obviously, that affected who asked to get into the trial and may have contributed to some of the exaggeration of symptoms by some patients who were enrolled. Frequency, in planning the 208 study, has addressed the mediating and moderating factors that influence studies. And based upon these lessons learned from 202, Frequency has applied the best practices to optimize the outcome for trial 208. I'm going to review some of these best practices. Frequency has developed a protocol that blinds the key eligibility metric criteria from both the site staff and subjects, something we call masking, and has employed multiple hearing assessment measures to distract the subject so that they don't know what the real eligibility criteria will be. These steps mitigate the risk of intentional score inflation of hearing loss deficits. The blinding addresses symptom misrepresentation, and the multiple measures make it difficult to falsify assessments. The blinding also mitigates the risk of site staff sharing specifics about the study because they simply don't know them. This can be done by employing a protocol addendum for the mask criteria that is not seen by the trial site. In addition, Frequency has added an interim visit between the screen and baseline visit to assess symptom severity and symptom stability. This addresses the issues of reliability, as well as symptom stability and symptom fluctuation. In addition, Frequency's added a rater training component to qualify raters and added site-independent monitoring by recording as a quality assurance layer. These steps address ratings precision and consistency through these surveillance measures. All of these steps ought to help. The result of all these measures, in my view, are going to optimize signal detection in this study, and I look forward to seeing the results. Thank you.
Thank you, Steve. As you can see, we have made significant progress in our hearing program. We thought it was critical to show all of our data, and we believe these data inform a clear path to developing the first therapeutic to restore hearing. Put simply, people's hearing does not improve. It only gets worse. In reviewing data from our studies, we believe we are already changing that paradigm for some patients. The people being enrolled in the 208 study represent millions of people like them who are motivated to treat their hearing loss. We believe FX-322 can be that treatment. But we're not stopping there. Chris Luce, our Chief Scientific Officer, first will introduce an exciting new program in auditory hair cell regeneration. It is our hope that this new asset, FX-345, will expand our ability to restore hearing for more patients and address more types of sensorineural hearing loss. But as I mentioned, we believe that our regenerative medicine approach has the potential to have a transformational impact in other degenerative disease areas. Sanjay Magave, our Vice President of Myelin Research, will unveil our first program outside of hearing loss, a novel preclinical remyelination effort aimed at repairing the underlying cause of MS. Remyelination is an essential area of MS research, as today there are no treatments to restore damage caused by the disease. We are very enthusiastic about the addition of these cutting-edge programs to our pipeline and believe they add significant value for patients and shareholders. We believe that our research may lead to treatments for these and other degenerative conditions in the future. I will now turn it over to Chris.
Thank you, David. I'm Chris Luce, co-founder and CSO of Frequency Therapeutics, and I look forward to taking you through two new programs that we are introducing today. At Frequency, we are working on a new way to reimagine regenerative medicine by reactivating the body's own regenerative systems. All of the tissue in our body originally arose from progenitor cells, and these progenitors often remain throughout life. In some adult organs, like the intestine, these progenitors are highly active, refreshing and restoring tissue every few days. But in others, they lie dormant. At Frequency, we are developing small molecule drugs that cue these dormant progenitors to reactivate and restore both structure and function. We call this approach progenitor cell activation, or PCA, and we believe this could have a significant impact on a range of degenerative conditions. As David alluded to, we see PCA as having three key advantages compared to other regenerative approaches such as cell and gene therapy or gene editing. First, there are no genetic changes made. We reactivate the body's natural genetic programs. Second, we harness the natural architecture of tissue, making the right cells in the right location. Finally, our approach has the benefit of small molecule manufacturing and drug delivery. This is particularly important in our first indications as we target the cochlea and the brain, which are challenging to access with biologics. In contrast, we can reach these targets with small molecules using standard and safe techniques for administration. As Kevin and Carl noted, this approach has been well tolerated and demonstrated a hearing restoration signal across multiple clinical studies. This led us to ask two key questions. First, what if we were able to get drug deeper into the cochlea? And second, how can we extend our PCA approach to other degenerative diseases where we could have a significant patient impact. Specifically, we've been talking about remyelination for multiple sclerosis over the last 2 years. Today, we will introduce data on a second clinical candidate in hearing restoration for sensorineural hearing loss, as well as a new program in MS. We have seen FX-322 produce a hearing signal when directly reaching the highest frequency region of the cochlea, as shown in orange. As Dr. Dar noted, improvements in this region could have a significant impact on speech perception. And we're excited to continue to advance FX-322 as the first potential hearing restoration therapy. And as David said, we believe that this has the potential to help millions of patients. Additionally, we asked ourselves if we could create a new candidate that directly reached a larger portion of the cochlea. And we have done just that. We have now created FX-345, a hearing restoration clinical candidate that achieves broad exposure through a large portion of the cochlea to assess if this could have an even bigger patient impact or help a broader population. As you also heard earlier from Dr. Dar, there's heterogeneity across hearing loss types. Deeper delivery into the cochlea using FX-345 may extend the reach of our approach. Further, FX-345 has greater flexibility in dose selection and formulation, enabling us to evaluate a range of different dose levels. While we need to bring this asset into the clinic to understand its potential impact, what we do know is that FX-345 may offer some advantages as we look to expand the opportunity to treat different types of sensorineural hearing loss. It's important to take a minute to review the underlying mechanism of action of FX-322 before discussing FX-345. FX-322 is designed to activate and proliferate cochlear progenitor cells, the first step in making a new hair cell. This progenitor proliferation is shown here in this FACS data, where you want to see more cells on the right. By using a single agent, a GSK3 inhibitor, which activates the Wnt pathway, progenitor growth is seen. A second agent, sodium valproate, is an epigenetic modifier. It inhibits HDAC. This means it opens up targets, but it on its own does not induce proliferation. However, the combination of these two agents, both opening targets and activating them simultaneously, gives a profound synergy, as shown here. This is the biology insight that led to FX-322 and our associated IP. Our delivery data showed the greatest opportunity for increasing effective cochlear coverage was to improve the potency of the GSK3 inhibitor. Now, I'd like to take you through how we achieved this to create FX-345 using a set of advanced tools that we have tailored for the new field of hearing regeneration. First, we built a discovery platform for evaluating the regenerative potential of a range of small molecule combinations in the right biological setting. Specifically, we use only freshly harvested primary progenitor cells from the cochlea in as native a state as possible. As shown here, these fluorescently labeled cells start as single progenitor cells, and drug candidates can be evaluated for their ability to activate and proliferate these progenitors into organoids. Using this cochlear progenitor screen, we identified a substantially more potent GSK3 inhibitor. We are building on the signal of FX-322 by combining this new GSK3 inhibitor with sodium valproate. Next, to evaluate the drug distribution, we've invested deeply in cochlear drug sampling and modeling. Specifically, we execute serial sampling in guinea pigs, allowing us to measure exact drug levels in different parts of the cochlea at different times. Further, we've worked with the leading academic to develop a computer model linking this preclinical data to human drug distribution in the cochlea. We validated this model by giving intratympanic injections to patients shortly before cochlear implant surgery, confirming that FX-322 reached the base of the cochlea, as we published in Auditory and Neurotology. We have used these preclinical cochlear PK studies and modeling to evaluate new approaches to achieving enhanced cochlear exposure. We have confirmed through cochlear sampling that in FX-345 preclinical studies, drug target levels are now reached through a large portion of the cochlea. Here we see human modeling data for FX-322 and FX-345 showing drug distribution levels across time and the location in the cochlea. Specifically, one axis is the position within the cochlea from the base to the apex. The second axis is the time from injection from left to right. The height of the surface is the concentration relative to the target level for the GSK3 inhibitor. As you can see, FX-345 achieves greater exposure through a larger portion of the cochlea for longer times. FX-345 is on track for an IND in the second quarter of 2022, which will be our first step in exploring the impact of broad and sustained cochlear coverage on a range of hearing loss populations. Now let's return to the second key question we've been pursuing in our research efforts. How can we apply PCA to create therapies for additional degenerative diseases? To this end, we've been developing a potential remyelination therapy for multiple sclerosis. As in the cochlea, the brain contains progenitors that require distinct signals to activate them. Our regenerative biology and bioinformatics teams have identified a novel target that is highly effective at driving remyelination from these progenitors, known as oligodendrocyte precursor cells. Further, we have developed multiple NCEs that are outperforming previously reported remyelination candidates in rigorous in vivo models. This continues to validate the PCA approach and has the exciting potential to lead to a new therapeutic option addressing the top need in multiple sclerosis, remyelination to restore structure and function. Now, our VP of Myelination Biology, Dr. Sanjay Meghavi, will take you through these advances.
Thank you, Chris. I'm Sanjay Meghavi, and I lead our discovery efforts for remyelination. Multiple sclerosis is a devastating neurological disease that affects nearly 1 million people in the United States alone. MS patients suffer from extensive demyelination in their brains, and since myelin is essential for neuronal signaling, MS robs patients of everything from their ability to see to their ability to walk, leaving them with profound, life-changing disabilities. As MS progresses, patients lose myelin and the accompanying oligodendrocytes. We intend to activate oligodendrocyte progenitor cells, or OPCs, to turn into oligodendrocytes and regenerate myelin lost to MS. There are both inflammatory and neurodegenerative elements to MS. While there are multiple drugs addressing the immune aspects of the disease, there's a significant unmet need for therapies that can remyelinate the brain. Our treatment, which could be used in conjunction with existing immunomodulators, will allow patients to regenerate myelin and may slow or even reverse disability in MS patients. When we reviewed known remyelinating agents, it became clear to us that we would need to develop something much more effective to drive meaningful patient benefit. We set out to discover new targets to generate a significantly better approach. We discovered a new target that we're really excited about. We can't disclose what the target is today, but our IP filings are in process, and I'm excited to tell you about it at some point in the future. At a high level, we combined our unique assays and expertise in progenitor cell activation with our bioinformatics resources to generate potential targets. There were a number of targets, but target 14 turned out to be the most promising. To validate target 14, we cultured OPCs in vitro and treated them with target 14 antibodies. The left image shows the controls, and the right image shows the treated cells. You can see a dramatic increase in myelin basic protein expression, shown in green, after treatment. Myelin basic protein is an important element of myelin that's expressed by mature oligodendrocytes. The graph on the right confirms that these results are highly significant. These results unequivocally demonstrate that hitting target 14 is sufficient to drive myelin protein expression in vitro. This discovery gives us a competitive advantage, since this is a novel, highly effective target that none of our competitors appears to be pursuing. We found multiple structurally distinct small molecules that hit target 14 and drive oligodendrocyte differentiation in vitro. We optimized one of these scaffolds to generate frequency 162. We have multiple compounds with promising characteristics, but I'm going to focus on 162 for the purposes of this talk. As you can see, 162 is highly potent, increasing OPC differentiation at single-digit nanomolar concentrations. 162 is orally bioavailable and also brain penetrant, which is essential for MS. Given these promising characteristics, we tested 162 in mice. As a first test of our compounds in vivo, we assessed whether they could drive oligodendrocyte differentiation. We stained for a protein that's transiently expressed as OPCs differentiate into mature oligodendrocytes. This protein is barely expressed in precursors, highly expressed in actively differentiating oligos, and turned off in mature cells. This allowed us to specifically detect newly generated oligos. This image shows a section from an adult mouse brain treated with a vehicle. Each of these bright red dots is a newly generated oligodendrocyte. We compared our compound to 3 known compounds from the literature. We dosed all of these comparators 3 times at or above the doses commonly used in the literature. The first comparator is thyroid hormone, or T3, which is widely considered to be the gold standard in literature. Thyroid hormone yielded an increase in new oligos. If you look very closely, you can see these new oligos, these little red dots in this image. The second comparator is an anti-LINGO antibody, a biosimilar to opicinumab, the antibody pursued by Biogen for remyelination. The third comparator is clemastine, which is an anti-muscarinic, a class of compounds being pursued by several companies. Both the LINGO antibody and clemastine showed very modest effects. Together, these are some of the most well-characterized and advanced compounds described in the literature. Now, let's look at the frequency compound. We administered just a single dose of 162 at 5 mg per kg. The frequency compound induces a profound increase in oligodendrocyte differentiation. We'd hoped for a robust improvement, but we still expected to have to spend days counting cells and running stats to show a difference. We were stunned when we put this under the microscope, and the difference was immediately obvious. When we zoom in, we can see individual new oligodendrocytes in the vehicle and 162 mice. Each of these cells has the classic morphology of a newly generated oligodendrocyte, with processes reaching out and searching for unmyelinated axons. 162 generated oligos in both the white and gray matter throughout the brain. Quantitation confirmed how dramatic these differences are. The graph on the right shows the number of newly generated oligodendrocytes throughout the brain. 3 doses of injected T3 drove a 6-fold increase in new oligos. A single oral dose of 162 yielded a 23-fold increase. These results show that 162 can induce robust, statistically significant increases of newly generated oligos. Thus far, I've been showing you that we can drive the formation of new oligodendrocytes. However, MS is a demyelinating disease, and patients need new myelin. To assess remyelination, we turn to the cuprizone model. On the left, you can see myelin basic protein staining in green in a healthy mouse. On the right, you can see a cuprizone-treated mouse. There are dramatic reductions in myelin in both white and gray matter. We want to really challenge our compounds, so we demyelinated these mice with cuprizone for 17 months. By the end of this process, the mice had lesions that were almost 1.5 years old. Further, the mice themselves were old and slow to heal. This is a much more stringent test than is usually used. We dose mice with the same comparators mentioned in the previous experiments, but for a longer time with up to 10 doses to allow myelin formation. The green staining shows myelin. The image on the far left is the vehicle. If you look very carefully, you can see a trend towards increased remyelination in the T3-treated mice and more modest changes in the anti-LINGO antibody in clemastine-treated animals. Now, let's look at the frequency compound. We administered just a single dose of 162 at 5 mg per kg. There's a dramatic increase in MBP in both white and gray matter throughout the brain. To the best of our knowledge, no one has published a remyelinating agent with this magnitude effect. The fact that we saw this robust effect after a single dose is promising and suggests we'll only need intermittent dosing to induce significant remyelination. When we zoom in on these results, you can see the details of the myelin and its location. On the left is the vehicle control, and on the right is the 162 treated brain. Cortex, gray matter, showed dramatic increases in myelination. This is important because cortical demyelination underlies the cognitive deficits that MS patients experience later in life. White matter tracts show remyelination as well. The lateral callosum and striatal tracts go from almost completely absent to clearly visible. This is important as white matter demyelination underlies the sensory and motor challenges MS patients experience. These data show that new myelin protein is generated in the right locations with the right morphology. I'll just quickly show you some results from multiple animals to demonstrate how consistent the response to 162 is. These are brain sections from every one of our vehicle animals and every one of our 162 animals from this particular experiment. Firstly, we get strong and reproducible demyelination with cuprizone treatment. Secondly, in every animal we treated, we saw a robust increase in myelin basic protein staining. We've tested 162 and our other lead compounds in mice in non-GLP pharmacodynamic studies at doses from 5 to 150 mg per kg over multiple days and have not seen any tolerability issues. Our understanding of the target gives us confidence that we'll be able to create a safe and effective drug. We quantitate these results, measuring MBP staining in the anterior forebrains of treated mice. On the left of the graph, you can see MBP levels in healthy control animals. Cuprizone induced massive demyelination. 162 yielded a significant improvement in myelination. Just a single 5 mg per kg dose brought MBP staining to nearly 40% of healthy levels. To summarize, Frequency has discovered and validated a novel target. We designed multiple novel chemical entities, including Frequency 162. Single-dose 162 induces robust increases in oligodendrocyte differentiation throughout the brain. Further, single-dose 162 induces significant remyelination, even in aged mice that have been demyelinated for almost a year and a half. Right now, we're advancing multiple candidates through essential studies to move this approach to the clinic. We've characterized the pharmacokinetic and metabolic properties of 162 and tested it in a variety of in vitro safety panels. With the favorable results so far, we're moving 162 to dose range finding studies. Other compounds are close behind. These data show the possibilities of progenitor cell activation for remyelination, and they illustrate the potential and power of the PCA platform for other diseases. Applying this approach to other serious diseases could yield life-changing new drugs. Thank you for your time. I'll hand it back to David for his closing comments.
Before we go into Q&A, I want to thank everyone for their time and attention. I believe our company has made some important advances this year, and I want to commend our team for their focus, vision, and execution, and most importantly, their commitment to bringing new regenerative treatments to patients. I hope that we have provided you a clear picture of our path forward today. In summary, we believe that we have a drug candidate for hearing restoration in FX-322 with clear patient response. We know the patients in which FX-322 has shown a response. We've learned from our previous trials and have designed a new trial with strong controls and FDA-approved clinical endpoints. We have a second new program in hearing that, based on the preclinical science, will lead us in a new and exciting direction. We have a novel regenerative program in multiple sclerosis targeting remyelination with a target that no one else has explored and with preclinical results that, thus far, exceed the gold standard in the space. And we have a strong, well-capitalized company and the resources to achieve the next set of milestones for these programs and deliver innovation and value for patients and investors. At the close of September 30th, 2021, we had unaudited cash of $160.5 million, which does not include restricted cash. Our balance sheet as of now can carry us into late 2023, and we will be able to advance the programs we discussed today to their next milestones. I'm confident in our team and our ability to execute as we continue our mission to transform the lives and standard of care for people impacted by a range of degenerative diseases. Thank you for attending today's R&D event. Our team looks forward to your questions. I'm now going to hand it over to Peter to moderate our Q&A.
Thank you, David, and good morning. I am Peter FREQ, Chief Financial Officer of Frequency Therapeutics. I will be your moderator for this morning's Q&A. Operator, we will now open it up for questions.
At this time, if you would like to ask a question, please press star 1 on your telephone keypad. Your first question comes from the line of Phil Nadeau from Cowen and Company. Your line is open. You may ask your question.
Yeah, sure. In terms of the data on the placebo in untreated ears falling outside of the 95% confidence, were the deficits in speech perception at baseline in the untreated ears the same as in the treated ears in the single-arm studies?
Bill, thank you for the question. Kevin, could I turn that over to you?
Thank you. When we looked at the data from all of the studies, we did compare the baseline scores and saw no differences grossly across the different trials. So we don't have evidence that that difference explained the data.
Perfect. And then second question on the FDA. Does the FDA have a preferred speech perception primary endpoint, or is the FDA relatively ambivalent between the different measures?
Carl, maybe you can address that question for Phil.
Yeah. Hi, Phil. In our conversations with FDA, it's the category of speech perception. You know that we're doing a number of different tests. Those tests are both in quiet background as well as noise background. And at this point, we're not saying which specific test is going to be the primary, but we're continuing to look, and that's what the 208 trial will tell us.
Got it. And then on the 208 trial, is it enrolling patients with moderate noise-induced hearing loss? It seemed like that group had a low responder rate in the prior studies.
Yeah, so the study has the upper end of mild and the lower end of severe. That's the range for severity. And then it's got both the sudden sensorineural as well as the noise-induced patients. And that's, in Kevin's section, that's where we've seen the signal primarily. So that's where we want to focus.
Great. And then one last question for Dr. Mugabe. Dr. Mugabe, very fascinating presentation. We're curious to learn a little bit more about the mechanism of target 14. Can you say maybe how it compares to the target of antilingo or some of the other targets that we're more familiar with?
Yeah, I'm really excited as a scientist to talk more about that target. But for obvious reasons, I can't really talk about it right now. We think it's a significant competitive advantage. It's sort of expressed kind of in the right places and in the right cells. I think that's what we can say at this point. Thank you.
Perfect. Thanks for taking our questions.
Appreciate the questions very much, Phil. Operator, who's next in the queue?
Your next question comes from the line of Anupam Rama from JP Morgan. Your line is open.
Good morning, Anupam.
Hey, guys. Thanks so much for taking the question here. Just two quick ones from me. So can you walk us through how the FDA became more comfortable with speech perception as the primary endpoint for FX-322, and from a regulatory perspective, how the novel PRO fits in? And then for the KOLs, what the importance of a PRO is from a patient perspective. Thanks so much.
So, Carl, hopefully you got that entire question, but maybe you can address some of those points.
Yeah, so in the interactions that we've had with FDA, I think, as I said in my section, there had been an expectation for there to be audibility improvements. But our data continues to point at speech perception as where we see the signal, and that's clearly why we've designed the 208 study with that primary. I think FDA is in agreement with that. We've had clear conversations with them. That's where the data is taking us, so I think we feel comfortable in that way. With respect to the PRO, it's still in development. We view it as important information that we think will help us understand what's the impact of FX-322 on daily activities of patients with acquired sensorineural hearing loss. And that'll be important for us as we start getting in discussions around reimbursement for the drug.
Appreciate the questions, Anupam. Don't know if you have any further questions to add to that.
Oh, I'm just saying, from the KOLs, the importance of the PRO endpoint from a patient perspective.
Sure, I can comment on that. Obviously, patient-rated outcomes have become very important across all fields and improve both engagement in the study and compliance with study procedures. I don't know how important it is in this study where we have a single treatment. On the other hand, this measure, exploratory measure, is uncoupled entirely from the primary measure. So it's not going to influence in any way outcome, if that's what you're addressing. So I think it's a useful exploratory measure. We'll see how it plays out. Long term, it'll be very useful in terms of clinical practice as a way of measuring treatment course over time.
Thanks for taking our questions.
Thank you very much, Anupam. Operator, we're open to take the next question in the queue.
Your next question comes from the line of Mayank Mamtani from B. Riley Securities. Your line is open.
Good morning, Mayank.
Hey guys, this is actually William. This is actually William on from Mayank.
Good morning, William.
A couple of quick, great presentation today. Just a couple of quick questions from us. We were curious potentially about how your enrichment standards for the upcoming studies have changed. How specifically you pull out those or reach those statistically and clinically significant patients.
Appreciate the question. Kevin, maybe you can address this one.
Sure. The slide we showed with the various circles that showed the response rate in different hearing loss severities and etiologies was an early guide to appreciate where we saw our strongest responders. We weren't necessarily limited by the strict definitions of mild, moderate, as the American Speech Language Hearing Association defines them. Rather, we looked to see where there were responses. And maybe, Carl, you can speak a bit more about the 208 inclusion?
Yeah, we haven't publicly disclosed, what the ranges are that we're recruiting in. As Steve Targum discussed, we're using a masked protocol. So for all the reasons that he described. And the sites know a particular range of hearing loss. That's on clinicaltrials.gov. That's 35 to 85 dB across a 4-frequency average. And that's how they recruit. And when they come in, then they get tested. And there are a series of other requirements for them to be able to qualify for the study. And that's what we're not disclosing.
Yeah. And Carl, maybe I can add a few things about the enrichment. I mean, and just step back a moment and think about Bob Langer's early comment about how drug discovery is never a straight path. A wise company is informed by their experience, and that's what's going into 208 in many ways. Some of the critical steps are adding multiple measures rather than a single measure as part of the eligibility criteria, and it's masked. Neither the patients nor the staff have any idea what it is, and they're doing it multiple times. By adding the interim visit, you can try to falsify your data. And, when you've had a novel treatment like this, you're creating new hair cells. That's exciting. Anybody with hearing deficits is going to want to get into this trial. But you don't know what the criteria is to get in, and you have to repeat that false criteria each time before baseline. And so we've really, I think, effectively masked it so that the patients who ultimately will be randomized will be close to getting us a reliable baseline metric. And as we said in the presentation, that's what it's all about. So I think we've done what we can to really mitigate the factors that can increase unanticipated placebo response.
Appreciate the questions, William. Not sure if you have any further additional points that you want to add for the team.
Yep, just two quick other ones. One for your FX-345. You noticed, you presented a very nice, nicely improved PK. I was curious if there was any other changes in that drug, whether with the gel formulation or different ratios, et cetera. And then just a second quick one on your MS-162 oral dose. Is that an oral dose? or what's your route that you're trying for?
Chris, I'll hand it to you first, and then you can hand it to Sanjay.
Thank you. Thank you for the question. The major change in FX-345 is actually using a more potent GSK3 inhibitor, and this is allowing us to achieve target levels of drug further through the cochlea, and we're excited to be able to explore this in the clinic to look at the impact of that change on patient outcome.
Sanjay, any thoughts on the MS front?
Yeah, just super quickly. It is an orally bioavailable drug.
Appreciate the question, William. Operator, we have about enough time for one or two additional questions, and then we'll wrap up.
Yes, sir. Your next question comes from the line of Paul Choi from Goldman Sachs. Your line is open.
Good morning, Paul.
Good morning, everyone. I hope you can hear me. There's a bit of an echo here. Two questions for us. First, as you think about 345, can you maybe comment on how you think about potentially the market segmenting between 322 and 345, or you think about co-administration or sequential administration here? And the second question is on 162. The histology that you've shown is very impressive. Can you maybe comment on what you've seen from a functional perspective in your in vivo work? Thank you very much for taking our questions.
Appreciate the questions, Paul. Chris, maybe you can kick it off quickly.
Thank you, Paul. First, I would emphasize that we're very encouraged about FX-322's signal we've seen across multiple studies. And the affected group that this would target that aligns with our trials represents 7 to 10 million Americans. And so we're very encouraged about taking that candidate forward as quickly as possible through additional studies, and we'll begin to study FX-345 in the clinic in parallel with that development. What we see in the clinical data will really guide us in terms of how we continue positioning those two.
Sanjay, any thoughts with regards to Paul's MS question?
Yeah, absolutely. So far, we've been really focused on discovering the drug, and we are just beginning experiments to look at its functional effects. So we're really excited to get those results and relay them to people.
Thank you very much, Sanjay. Operator, I just want to thank everyone for joining today's R&D event. From David, myself, the rest of the management team and our company, we appreciate everybody attending today's event, and we look forward to talking to you in the very near future. Thank you again.